Thanks. Thanks everyone for attending. So yeah, I'm going to talk about how we use our synthetic DNA technologies to discover antibody ligands for GPCRs. So Twist is a DNA synthesis company. So just as an overview, I want to talk about how we leverage DNA synthesis for antibody discovery using phage display and synthetic libraries. So we're a small team within Twist called Twist Biofarma. And then I want to go through 3 case studies where we've had successes. So the first is a GLP-1 antagonist, not an agonist. So this has implications for hyperinsulinemia, which is a rare disorder. So we found an antibody that is 100-fold more potent than the current standard of care for hyperinsulinemia. And using our synthetic libraries, we were able to pull apart some of the mechanism of activation of GLP-1R. The second, I wanted to ask the question if we could find unique pharmacology within our libraries. So I thought the APJ system was really nice for that because if you activate APJR, it's a treatment for heart disease and other cardiovascular disorders. If you block it, it's an effective treatment for cancer. And the third story is about the C5A receptor. And we have a really cool story here where we look at biased agonism of the receptor and take it all the way to primary immune cells. So the DNA synthesis platform at Twist is this chip about the size of a 96-well plate where you can print 1 million oligos on the chip. So we have high quality, speed, low cost oligo synthesis. And what we do is build synthetic phage display libraries. So a normal phage display library typically uses degenerate PCR to give you random variations. For us, we design in every variation. So we synthesize every oligo that is in our libraries of 10 to 13 molecules. So there's a lot of advantages to using printing libraries, which are outlined here, but I think the biggest one is we can vary the CDR length within one library. And I probably don't need to go into too much detail here, but this is one arm of the Y of an antibody, which will have 4 CDRs. So our oligos are, the oligos we synthesize are again designed variations of each CDR, with special emphasis on CDR3, and we remove liabilities and also design specific epitopes that bind to cryptic sites. Again, more background. So this is the process of phage display. So going from synthesis to the library build, you put the library into phage, so it expresses the single chain variable fragment, and then you bind it to cells or purified proteins expressing your receptor of interest. Then we basically wash, purify, go through this process 5 times, and then do next-gen sequencing to collect our specific antibodies. So just to talk about the CDR3 design I was alluding to, this was inspired by this paper from 2014 from Scripps. What they did was they took a peptide antagonist of CXCR4, this is called CVX15, and they fused it to a bovine CDR3. So the bovine CDR3, this BLV1H12, is extra long, so it could stretch out from the antibody and fit into the ligand binding pocket of CXCR4. So we were sort of inspired by this, and we took 5,000 peptides that were known to bind GPCRs, so basically 5,000 peptides that would bind inside the binding pocket, and we fused them into 5 different ultra-long CDR3s from humans. So this created a library of 150,000, different CDR3s and ligands in the library, and then when you do the combinatorial mixing of the other CDRs, you get a 10 to the 10 pages per library. So this is just to show you the CDR3 length of our GPCR library versus human B-cell repertoire. They're all very long, over 20 amino acids. And then this is an AlphaFold model we did. So this is the SCFE, and then you can see stretching out here is a fragment of the GLP-1 peptide. So that's here, that's the GLP-1 peptide. So at Biofarma, we set up this 6-step workflow. I talked to you about the phage libraries, cell panning, sequencing. We reformat from phage into a full-length IgG. We have a high throughput IgG purification platform. And me and my group, we work on doing the binding and functional assays. So again, I want to tell you 3 stories: the GLP-1 story, the APJR story, and the C5A story. So GLP-1 is overexpressed in XP293s. We use purified APJR and proteoliposomes, and then just overexpressed C5AR1. And then for GLP-1, I did high throughput binding and screening in parallel, and then took this all the way to in vivo studies in mice. So I talked to you about how we found this original GLP-1 antagonist from the GPCR library. We have another platform for affinity maturation we call Twist Antibody Optimization. So here we're building a whole library around this one molecule that has high affinity for GLP-1R. What we're essentially doing is trading out CDR1 and CDR2 and the light chain CDR3 for CDRs that are in the human repertoire. And then we're essentially making point mutations of that GLP-1 peptide in the CDR. So there's 433 mutations, and we have an entire library now, starting over from the panning, that will hopefully give us an optimized antagonist for GLP-1R. So after the whole synthesis and panning process, we scaled up the top 94 binders and did binding and signaling assays, and we found 11 optimized antagonists and actually 2 partial agonists where the antibodies switched into activators of the receptor. Sorry. So this is like my favorite part is looking at the receptor activation through G protein activation, transcriptional activity, and beta-arrestin recruitment. Traditionally, for an antibody screening campaign, you look at binding at equilibrium, and this is your triage for the best antibodies, but I wanted to, in parallel, do G protein activation inhibition and also arrestin binding. The reason is, a lot of ligands and agonists, they don't necessarily have the best binding profile, but they could be better drugs down the line. So, yeah, that's essentially this bar graph here is looking at all 3 activities downstream of the receptors. So, just to get into more detail about this optimized GLP-1 antagonist, we're calling 22223. This is that same AlphaFold model in blue. Now it's aligned to the structure of GLP-1 bound to GLP-1R. So it's not actually in the cryptic binding site, it's replacing GLP-1 and the N-terminal binding domain. This is just the flow cytometry. You can see it binding really well over background, and also in this graph, you have the binding versus the expression of individual cells of the GLP-1 receptor. So for antagonism, the current standard of care for hyperinsulinemia is extended 3, and you can see our 2 antibodies have about 100-fold left shift better blockers that G protein blocking. Something that's kind of interesting for the beta-arrestin pathway, all 3 antagonists are basically on top of each other. So this is an improved antagonist over extended 3. with our collaborators in Philadelphia, we looked at a mouse model of hyperinsulinemia. So this is a transporter that's mutated in genetic order disorders. So these mice have higher levels of insulin. You block GLP-1, you lower the insulin levels. So the first readout here is treatment with our antagonist increases plasma glucose in a glucose challenge test. And then the next step is we isolated islets from pancreas, and we looked at insulin release in these islets. So when you treat with our antagonist here, you see no insulin release with the stimulation. We also did the same assay with a patient that has this mutation, and you can see the same blocking effect with our antagonist antibody. So again, this disorder is treated with exendin 3, and we have an antibody which has a better half-life and better pharmacology for the receptor. As I alluded to, our phage display, our antibody optimization was essentially point mutations of that GLP-1 peptide. So in doing the functional screening, I found some really interesting readouts. This 22289 is just a mutation here in that very C terminus of the peptide, and 22240 is another point mutation. Basically, these change from being antagonists to very weak partial agonists. And actually, exendin 3 is also, it's a weak antagonist and also a weak partial agonist as well. So it's really cool that you can activate the receptor by just pushing on that N-terminal domain with these different mutations. Okay. So the next story is about APJR. Again, doing binding and function in parallel is really the goal here, to see if we can pull out agonists and antagonists from our library. So just as brief background, this is a rat model of heart disease. They were treated with an APJR agonist, and it alleviated these phenotypes in rats. And then APJR is very well established to cause vascularization when it's activated, so blocking it is a really potent way of causing tumor death. So again, the big question is, can we pull out functional antibodies that act as agonists or antagonists at APJR? So again, this was panning against proteoliposomes with APJR. Here's agonist mode for just the flow sensor cyclocane P assay. So here's the control ligand on this axis. Remember, APJR is GI coupled, so lower is active and higher is blocking. On this axis is the binding. So you can see the top binders are not necessarily the best. They don't end up being the best agonists or antagonists. So there's three interesting leads here. I just want to pull out the dose response curves. So on the top is G protein blocking, the curve is going up, and on the bottom is beta-arrestin blocking for this H116. Here we have activation of the G protein pathway and partial activation of the beta-arrestin pathway. And I think this is really cool. We're pulling out leads that really don't do anything on the G protein pathway but activate the beta-arrestin pathway. So partial agonists, or biased agonists. Okay, this is a little more fleshed out. This is the C5A overexpressing cells. I took this through this high throughput screening and functional assays and brought it all the way to primary immune cells. C5A is really interesting, and there is a lot of interest in targeting C5A with antibodies. Biofarma has a molecule that they've looked at in clinical trials for anti-inflammation as well as immuno-oncology. So we'll get to that later. And then there's one small molecule inhibitor, C5A, called the which is used for an autoimmune inflammation disorder. Just looking at this from a high level, it's really interesting that The C5a receptor, the C5a peptide can stimulate the immune system systemically, but then also suppresses the immune system in the tumor microenvironment. So again, if we have all of these antibodies that bind with really unique pharmacology, can we better dissect these two sort of paradoxical activities in the C5a receptor? So again, looking at activation, this is another GI coupled receptor versus binding with agonism and antagonism. I think what I want to point out here is none of these were very strong activators or blockers. When I went back and did functional assays, here this 124-105 did seem to activate, but it was really right shifted from the endogenous agonist. There's really no blocking of the receptor, but it did have very potent beta-arrestin blocking. That's this red line versus the toperous small molecule in this purple line. So it seems to be a beta-arrestin biased antagonist. This other one is kind of clear why it's in the middle here. It's a partial agonist. So it activates and blocks the G protein pathway and the beta-arrestin pathway. Here you just see the blocking and activation curves coming to the same point. I wanted to understand really the mode of binding. So I did the binding, and you do see weak binding, and you titrate in the peptide C5a for that partial agonist. There's really no blocking of the binding, but this beta-arrestin biased antagonist really gets knocked off of the receptor. So it seems to be competing for the peptide binding site, while the other one is an allosteric modulator. And then we worked with G-clips that gave us some really nice solubilized C5a receptor to do direct binding for this 105. So it does, even though the binding is weak, it does bind to the receptor really well. Okay, so the last little piece here is just the functional assays. So here we just have activated PBMCs. C5a receptor is on monocytes and is known to inhibit NK cells through this pathway. So when we block the C5a receptor, we see activation of the NK cells. So here you can see that small molecule, which is pretty weakly effective, is having activity in this assay. Here, the partial agonist is actually looking, overlapping with the small molecule, and then a beta-arrestin biased antagonist is really the best blocker in this assay. Second assay, I just wanted to look at calcium flux, sort of a proxy for inflammation. So just bathe the PBMCs with C5a and look at the immediate calcium response. So that's here. You can see the dose response for calcium flux C5a, and this 124-105 blocks that calcium response. In this case, the partial agonist really doesn't activate the calcium response or block it in these total PBMCs. So yeah, that's the whole story, just to summarize. We're doing parallel binding, functional screening of this really rich antibody library that's targeted specifically for GPCRs. I told you how we pulled out a very strong antagonist and also used the library diversity to sort of figure out the activation mechanism of GLP-1R. Then I showed you how we can sort of pull out agonists and antagonists for receptors of interest, and also how we can let the interesting pharmacology sort of drive to the preclinical models. So yeah, I think Aaron, who leads our team, and then my RA Cameron, who helped me with a lot of this work, and then the rest of the team with the antibody preparations, and of course the panning too. So, yeah. Thanks.
So I can start with the first question here. So I know, antagonism and agonism can be modulated by isotype, at least with other receptor classes, and you have so many functional antibodies. I'm just curious, have you explored isotype or hinge flexibility modulation of your antibody activity with all this interesting chemical matter you have?
Yeah, so they're all in IgG1, and we switch, depending on the endpoint, we'll switch to IgG4 to not have that FC effector function, but then also we do see like enhanced activity when there's more flexibility. So it's really just, it's something we do, probably the next step after this functional characterization. So yeah,
Thanks for the nice talk. I had a question about the speed compared to immunizations and so on.
Yeah.
Is the synthetic library taking a long time to generate, or?
So we sort of have it all on the shelf. So I have that card with the different libraries. So as far as like the generation, it probably takes like 4 weeks to do it de novo, but for most of what we do, we have, like half a dozen libraries, different libraries, including that GPCR library.
Thank you.
Hi, did you use the GPCR library for all of these targets? And do you consider using, say, your antibody library for class A GPCRs that have less surface area?
Yeah. So this is all the GPCR library. Typically, we actually mix 2 libraries or do separate library panning arms together.
Do you have any plans in putting the same like GPCR library into the VHH scaffold and not just the IgG scaffold?
Yeah, we have that. We just got it online this year. Yeah, I'm really excited about that.
And then for your GLP-1 Fab, where you were you said there were like 400 mutations that you made in the CDR3. Yeah. Was that just every possible mutations? Because I didn't know how many residues it was, or did you use like a structure or like degenerate codon scanning to figure out what you wanted to mutate it to?
Honestly, I don't know, but the peptide is like 10 amino acids. Okay. So I think we probably did like
everything?
Okay. Yeah. And then probably different CDRs. Within that CDR3, grafted CDR3, there's differences there. So.
Thanks.
We have time for additional questions if anyone has anything.
You also said that you for your libraries you can also change the length of the CDRs.
Yeah.
So yeah, what like how long do you go till and how short are they?
Yeah, I don't know. When you get super long, like over 40 amino acids, it's just way too unstable. So we've made libraries like that, and they just like don't express when we reformat into IgG. So I have the graph here that just shows for this particular library, there's 2 peaks. Most of them are around 30 amino acids, and the other peak is around 40. I think. Yeah, so that's the CDR length and the distribution. So that's a histogram distribution in this library of 10 to the 10.
Okay, I guess if there are no more questions, we can wrap up and thank the speakers from this panel. We'll reconvene at 3:40, so please join us for more presentations at that time.